Okay, chalo. So let's learn about gram staining. So gram staining was discovered by, this technique was developed by, this practical procedure was developed by a scientist called as Christian Graham. Okay. Then second, very important thing we got to remember in this is it is differential staining technique. Differential staining technique means what? We can differentiate two different type of bacteria based on its, we will follow same procedure, but ultimately during result we get two different colors, two different bacteria with two different colors. Understanding? So it is called as a differential staining technique, right? So now let's go to the further part. Now let's see the procedure of the experiment first. Okay. Procedure of the experiment first, and then we will go and see why we get two different type of bacteria. Okay. Is screen visible?
Yes.
So see this diagram. One second. This is a procedure of it. We will copy it and attach it. So see over here. This explains the procedure of the experiment. Okay. Now, what we do? See, first step of the experiment is that we need to take bacteria. What we need to do first? Take bacteria. Okay. Now, take bacteria, I am saying here for you to understand, but scientifically we call this as a preparing bacteria or preparing slide. Right. So what we do? See, in school you will be provided with a in small vessel you will be provided with some bacteria, some solution, liquid solution. You take a dropper. Okay. Take one drop of that liquid, okay, जो provide किया हुआ, like whatever provided liquid, take one drop onto the slide. Understanding? Take one drop onto the slide. Then on that drop, you put something called as cover slip. Okay, a circular small glass thing is given called as cover slip. Cover slip just allow us to what to say, allow us to fix the bacteria at one location. See, then afterward we understand that my bacteria are over here. That's it. Okay. Sometimes we put cover slip, sometimes we don't put cover slip. It depends. Then what I need to do is I need to heat this. I need to heat this. So because of heating, what happens? This bacteria, which I have put onto the slide, they get stick to the slide. They die. That's true. They do not remain alive, but they get stick to the slide. Understanding? And water get evaporated. Fine. Then I can remove cover slip. After this, what? Remove cover slip. Okay. Remove cover slip. Right? Okay. Now your bacteria are said to be fixed on slide. Fixed on slide. I fix them by heating. Understanding? Then the second step. You understood first step? First step is clear?
Yes.
I just need to fix my bacteria onto the slide. Okay. Then I add a stain. Stain you understand? Stain you understand? Vaibhavi? Stain matlab color. Stain is what? Color. Okay. See, if something ink or something fall onto your dress, what you say? My dress is stained, correct? Understand? So here stain is color. Okay. So color called as crystal violet. What is the name of color? Crystal violet. It is a name of color, right? This crystal violet, I add on to my bacteria, right? Now, as it name, it's very violet colored, very dark violet colored stain, right? So whole my slide become violet colored, right? Particularly where the bacteria is, no, that area become very much violet color. Then what I do, I rinse my slide with the water, right? I rinse my slide with water. But I need not to rinse it under the tap. Otherwise, bacteria will go away, correct? I need to take water, little amount of water in a beaker, and I need to gently, gently wave my slide inside it. So extra color will go on. Okay. Then make the slide dry again. Water जाने दो उसपे जो भी है. Okay. Then see, wash extra color. See, my first step is fix it. Second step is add stain. Third step is wash slide to remove extra stain. To remove extra stain. Right? Extra stain gaya abhi, chala gaya. Now make it dry. Make the slide dry. Slide dry. Okay. Once it get dry, now add next stain. Now add next stain called as gram iodine. Add 1 to 2 drops of something called as gram. This person gave his own name for that. Gram iodine. Means basically we are adding iodine. Okay. Now iodine is something called as mordant. मतलब iodine क्या करता है? Try to understand. It fixes the stain, fix the color, fix the crystal violet color to the bacteria. Fine. Crystal violet color to the bacteria. Fine. It fixes the crystal violet color to the bacteria. Understanding? Okay. Now bacteria took, because of iodine, bacteria take the color and it won't leave. Understanding? Fine. Then again wash the slide to remove iodine. Okay. Again wash the slide to remove extra iodine. Wash the slide to remove extra iodine. Slide to remove extra iodine. Extra iodine. Then again, okay, again wash the slide or rinse the slide with alcohol. Rinse slide with alcohol. Okay, with alcohol, little bit of alcohol. Rinse the slide. Again wash the slide with, to remove alcohol. To remove alcohol. Understanding? Okay. Remove alcohol. Fine. See, we will repeat steps by the way. We'll repeat steps. See, first step of experiment, what we need to do, we need to take drop with the help of dropper. I need to take little amount of bacteria onto the slide. Okay, but along with bacteria, I took water also, and the bacteria may be swimming in water right now. Correct? Okay, so I need to remove that water and I need to stick my bacteria onto the slide. So what I did, I heat it and water get evaporated. I placed cover slip just in case, because you students are new, no? That's why you guys put cover slip for a while. Okay, but afterwards you remove that. Right? So you heat it, bacteria get stick to it, water get evaporated. Okay? Once water get evaporated, you added crystal violet color stain. Correct? Very violet color stain you added. Okay, but when we add, we always add extra. So we need to wash it. So wash extra crystal violet with water. Okay, dry it, add iodine to it. Okay, iodine क्या करेगा? Iodine will fix the crystal violet color. ठीक है? Iodine will fix the crystal violet color. Again wash it to remove extra iodine. ठीक है? Then next, after adding iodine, wash it again to remove iodine. Okay, then make it dry. Again add alcohol to it, little bit. Okay, wash that alcohol with water. Right? Now again add. Right? You wash extra alcohol. That we, up to that we have studied. Is that clear? Is that clear?
Yes.
Now.
Yes.
Remember steps, huh? Remember steps very, very carefully. एक बार अल्कोहल से वॉश हो गया. Now add Next, second stain. Now we need to add second stain, and second stain is called as saffron. Now add saffron. Saffron, add saffron into it, and then again wash extra saffron. Wash extra saffron in with water. Extra stain, saffron in again stain, color. Okay, saffron is a pink color stain. Crystal violet was a violet color. This is a pink color. Okay, extra stain with water. With water, make it little bit dry a little bit, and now observe under microscope. Observe under microscope. Right? This is my procedure of the experiment. Understanding? This is procedure of my experiment. Is that procedure clear to you? Remember, I added two stains. A crystal violet, first stain, then iodine just to fix that crystal violet, and later on I washed stain with alcohol, and afterwards I added saffron, another stain. Correct? Okay. Saffron is a pink color or violet color. Okay. Suppose you have taken as a sample, initially जो bacteria का sample लिया था, suppose we took a bacterial sample from curd. Okay. Suppose our sample was curd. All bacteria which I will see are pink colored. Okay, because they will just stain by saffron. Okay, I'll tell you what happened to them. Suppose I'm observing curd, it will be all of them will be pink colored, right? Because the bacteria which make curd Vaibhavi, they are pink colored, they are gram negative bacteria. I will teach you that a little while. Okay, then but suppose I have taken bacteria from the root of the root nodules. You understand root nodules? जो nitrogen fix करते हैं. Nitrogen fixing bacteria. Have you studied nitrogen fixing bacteria?
No, ma'am.
No, not studied. Okay, they are like leguminous plant जो होते हैं. Like leguminous plant means the plant which produce legume. Legume means shenga. Whatever plant produce, legumes. Those plants are having special bacteria in their roots. Okay, those bacteria are called as nitrogen fixing bacteria. We will learn about them. Actually, we had that topic, mineral nutrition, but they deleted it, unfortunately. But if we have that in state board, we will study them, study that topic anyhow. Okay, so those bacteria are suppose I took a sample from the root of the plant, then I will get violet colored bacteria. Okay, I get violet colored bacteria. Okay, so first of all, try to understand why we get two different colors of, like some bacteria, why I get violet in gram staining, some bacteria why I get pink in gram staining. Let us try to understand it. Okay, क्या होता है exactly? What exactly happened during the gram staining procedure? I want you to remember procedure, huh? Remember procedure properly. Okay, so remember, see, you can see two type of bacteria over here in this particular diagram, in this particular representation. एक है ग्राम पॉजिटिव, एक है ग्राम पॉजिटिव बैक्टीरिया. ये स्टेन होंगे पर्पल कलर्स. These bacteria will be purple in color, and another are gram negative bacteria which will pink in color. Fine. Now scientists, after discovering this technique, they were wondering why some bacteria pink color and some bacteria remain purple color in this procedure. Okay, so let us see what happened in each step. Okay, so first let us see what happened, why some bacteria remain purple in color. See, I have taken a bacteria onto the slide. I fixed them. Agree? Now, when I added crystal violet, they become purple color. Right? Crystal violet, they become purple color. Okay. Then I added iodine. Purple color is fixed. Right? Purple color is fixed. Agree? Then what I did, I washed it with alcohol. Right? washed with alcohol. When I washed with alcohol, आइडियली क्या होना चाहिए था, ये पर्पल कलर वॉश आउट होना चाहिए था, बट ग्राम पॉजिटिव बैक्टीरिया पर्पल कलर वॉश ऑफ नहीं होता अल्कोहल से, राइट? द पर्पल बैक्टीरिया रिमेन पर्पल. Clear about it? So when we observe
Yes.
under microscope, they appear purple. ठीक है? Under microscope, they appear purple only. On the opposite side, पिंक कलर की बैक्टीरिया में देखो क्या हो रहा है, ग्राम निगेटिव बैक्टीरिया में देखो क्या हो रहा है. ओके, I fixed the bacteria as usual, correct? Then I added crystal violet, bacteria become purple. Agree? Then I added iodine, purple color is fixed, fine. But when I washed with alcohol, the purple color washed off. Understanding? The purple color washed off. Okay? Crystal violet stain washed by alcohol, right? Stain get washed with alcohol, right? And then as purple color get washed off, they again become colorless. Agree? And then I added safranin, right? Safranin was a pink color stain. So as they become colorless, they achieve or they took up this pink color. Understanding? They took up this pink color. Clear about it?
Yes.
So, what to say? So, gram negative bacteria appeared pink and gram positive bacteria appeared purple in color. This is what happened. Now the next question is why? Why few bacteria do not lose their color after washing with alcohol, and why few bacteria lose their color after washing with alcohol, correct? So when this Christian Graham, when the scientists studied those bacteria in detail, he found out that there is a difference in the arrangement of cell envelope in both the bacteria. मतलब ग्राम पॉजिटिव बैक्टीरिया का सेल एनवेलप अलग होता है. इट इज डिफरेंट देन द ग्राम नेगेटिव बैक्टीरिया. ओके, लेट मी शो यू हाउ डिफरेंट इट इज. ओके, इट इज डिफिकल्ट, Vaibhavi, difficult to understand?
No, ma'am.
नो ना. सो ये देखो, ये है सेल एनवेलप ग्राम नेगेटिव बैक्टीरिया का. ओके, एंड ये है सेल एनवेलप ग्राम पॉजिटिव बैक्टीरिया का. ग्राम पॉजिटिव रिमेन पर्पल, राइट? रिमेन पर्पल and this become pink, correct? Pink, lose its color. Okay, so in gram positive bacteria, as you see, this is cell wall. This is what? Cell wall. And remember, in case of bacteria, cell wall is made up of peptidoglycan. Remember this chemical name? Peptidoglycan.
Yes.
Okay. So if you can see, there are many layers of peptidoglycan in gram-positive bacteria. Okay? Many layers of peptidoglycan, and below that, plasma membrane, right? Plasma membrane. So when I wash this bacteria with alcohol, peptidoglycan को कुछ नहीं हो रहा है अल्कोहल से. Nothing happened to peptidoglycan by alcohol. But plasma membrane जो होता है ना, plasma membrane dissolve in alcohol. Right? Okay, but as there are multiple layers of peptidoglycan, alcohol do not reach to the plasma membrane, right? Alcohol do not reach to plasma membrane. Understanding? So it gets protection because of thick layer of cell wall. Understanding? That's why उनका color lose नहीं होता. Gram positive bacteria, right? Clear?
Yes.
So suppose Vaibhavi, we got infection of gram positive bacteria. It is difficult to cure क्योंकि उसके अंदर कुछ भी penetrate नहीं हो रहा है easily, right? Nothing is getting penetrated easily. Very, very difficult. Agree? Difficult. But on the other hand, gram negative bacteria देखेंगे, so you can see only one peptidoglycan, correct? Thin layer of peptidoglycan, correct? And there is a plasma membrane above, there is a plasma membrane below. So when we wash it in alcohol, this plasma membrane get washed off. Fine? It become disappear. Agree? Understanding?
Yes.
It get disappear and alcohol get easily penetrated. So crystal violet stain get washed away. Understanding? Okay, it get washed away and it lose its stain. So वो अपना color भी lose कर देता है. It become colorless. And when we add, when we add safranin, it take pink color. Fine, complicated to understand, right? Little complicated, right? So you need to study this once again. See, remember? You must have understood. I know. Okay. But just difficult to recollect. जब question आता है तो turn करना थोड़ा difficult हो जाता है. Fine. Little become difficult during the examination time of point. Okay. So remember, few bacteria you remember. बैक्टीरिया जो कर्ड वाला बैक्टीरिया होता है इट इज ग्राम नेगेटिव और नाइट्रोजन फिक्सिंग बैक्टीरिया जो होता है वो ग्राम पॉजिटिव होता है. फाइन. इजी.
Yes.
Yes. Okay. So our gram positive, gram negative things are over. Okay. So the prokaryotic cell is over. Just I forgot one thing to tell you that I will tell you about prokaryotic cell and we have completed prokaryotic cell today. Okay. एक चीज मैं भूल गई actually during when I taught you ribosome. Okay. See, grandstanding is clear now. No confusion in that now.
No.
No, sure. Okay. Just I forgot one thing. Just let us go back a little bit to our this ribosome. Okay. I forgot to tell you. That's why I'm telling you. Now I just looked at the book and I realized I did not teach you that. One second, huh? हाँ क्या है वो ultrastructure. One second, huh? हाँ. See, Vaibhavi, I taught you this. I taught you. Where it is ribosome, correct? Over here. So I told you ribosomes are present in cytoplasm, correct? Ribosome cytoplasm में होते हैं. I'll just draw bacteria so it will be easy for us to understand. Okay, so in bacteria देखो क्या होता है. Okay, this is a bacteria. I'm just drawing a simple structure. This is a bacteria. Here it will be its DNA. Okay, and ribosomes will be here. They are a small dot structures, correct? But larger subunit, smaller subunit, larger subunit, smaller subunit. And ribosomes do the process of protein synthesis. Ribosome क्या करते हैं? Ribosome do the process of protein synthesis. Okay, protein synthesis. Okay, about protein synthesis procedure, no, we have extremely detailed procedure next year, 12th standard. But right now you just understand when these ribosomes do the process of protein synthesis ना वो एक साथ आ जाते हैं. एक है राइबोसोम, उसके साइड में ही दूसरा आ जाता है, उसके साइड में ही तीसरा आ जाता है. फाइन. ओके, उसके साइड में ही फोर्थ आ जाता है. ओके, उसके साइड में ही फिफ्थ आ जाता है. फाइन. And likewise they arrange themselves and everyone is making protein. They work like a protein factories. Understanding? Okay. Understanding how they arrange themselves? Clear?
Yes.
So whenever we observe them under microscope, okay, we see there are many ribosomes attached, okay, to each other. Okay, and we see a very long chain of ribosomes. We see very long chain of ribosomes. Very, very rarely we see one ribosome separated. Otherwise, most of the time we see this chain. And this chain of ribosome is called as poly. Poly means many. Polysomes are polyribosomes. Polyribosomes. Right? Polysomes are polyribosomes. In bacteria, we see mostly polysomes or polyribosomes. Clear?
Yes.
So this I forgot to tell you. You add this point in your ribosome thing. Okay? Okay, we will stop this recording. It's enough.